let's spend some time reviewing the components and the organization of the autonomic nervous system. This is also done in gross anatomy, but there are going to be a couple of additional features that will be relevant, particularly to neuroscience. First point is the functions of the autonomic nervous system. It is exclusively a motor subset of the nervous system, and it's responsible, as we say, for the motor innervation of all smooth muscle in the body, whether it's vascular smooth muscle, gut smooth muscle, the heart, and all of the glands of the body are under ANS control. And as we know well, the autonomic nervous system has two separate, somewhat antagonistic subdivisions known as the sympathetic and parasympathetic subsets of the ANS. Common feature of both sympathetic and parasympathetic pathways is that in the peripheral nervous system, they utilize two neurons to reach their target tissue. And the diagram that illustrates that is shown on the next picture, whereby in all autonomic pathways, all preganglionic autonomic neurons will have their cell bodies somewhere inside the CNS. Their axons will leave in a spinal or a cranial nerve, and that preganglionic axon will synapse somewhere in an autonomic ganglion, which we know is derived from neural crest cells. It will be then the axon of the neural crest cell derivative ganglion, which will branch out and reach the target tissue, regardless of whether it is smooth muscle, a gland, or the heart. Now, we know descriptively that there are a number of different kinds of neurotransmitters and receptors associated with autonomic pathways, and that's kind of illustrated schematically on this slide. Note first off the very different feature about how skeletal muscle is receiving its innervation. Skeletal muscle basically is being innervated by a single motor neuron whose cell body is somewhere inside the CNS and whose axon leaves in a spinal or cranial nerve and basically utilizes acetylcholine at a nicotinic receptor to make skeletal muscle contract at a neural end plate. In contrast, as we know, all autonomic pathways innervating glands, smooth muscle of the heart are two neuron pathways. And we make the generalization that all preganglionic neurons, like motor neurons that innervate skeletal muscle, utilize acetylcholine as their neurotransmitter, regardless of whether they are a sympathetic preganglionic neuron or a parasympathetic preganglionic neuron. Then the postganglionic neurons are going to be a little different. All postganglionic parasympathetic neurons also utilize acetylcholine that interacts with muscarinic receptors. Sympathetic neurons have a variety. Many postganglionic sympathetic neurons utilize norepinephrine that interacts with either alpha or beta adrenergic receptors in various organs and tissues. Just sweat glands and the piloerector muscles are cholinergically innervated, even though they're under sympathetic control, and they're interacting again with muscarinic receptors like the parasympathetic neurons. And finally, as we illustrated, neural crest cells will migrate into the adrenal medulla, in essence forming a sympathetic ganglion that is releasing mainly epinephrine with a little norepinephrine by way of neurohumoral transmission. Next, here's a summary again of what we just said. Major difference, which we'll see, is that a somatic motor neuron is considered to be a motor neuron that's involved in the innervation of skeletal muscle. And that is simply a single neuron pathway, cell body in the CNS, and an axon that reaches the neural end plate of the skeletal muscle. Autonomic nervous system is exclusively a two-neuron pathway, where I need a pre- and a postganglionic neuron to interact synaptically in order to reach a target tissue. And the other point that's made on here, and we'll see this diagrammatically in a minute, is typically, but not exclusively, preganglionic parasympathetic axons tend to be very long. Preganglionic sympathetic axons generally are much shorter. And the reverse is true of the postganglionic. So parasympathetic pathways have a long pre and a short post, whereas sympathetic pathways typically have a short pre and a long post, but there are going to be some exceptions to this, as we will see. Next, let's look at the overall organization first of the parasympathetic nervous system. Note first that the location of the preganglionic parasympathetic neurons have a craniosacral distribution, meaning that the preganglionic parasympathetic neurons will leave in one of four cranial nerves, the cranial distribution, or their axons will leave in the ventral roots of sacral spinal nerves. So if we expand this diagram and cut it into two pieces, we can get a little more detail about the organization. First, looking at the cranial component. Note that there are four cranial nerves that carry preganglionic parasympathetic axons out of the brainstem. They are cranial nerves 3, 7, 9, and 10. Cranial nerves 3, 7, 9 just innervate glands and or smooth muscle that's located in the area of the head, whereas the vagus nerve parasympathetics are not distributed to head and neck viscera. They're being distributed to thoracic and most of the abdominal viscera in terms of heart, lungs, and GI tract. So being specific, and we'll deal with this when we review the cranial nerve anatomy in its entirety, cranial nerve 3, through a synapse in the ciliary ganglion, just innervates two smooth muscles in the orbit, the sphincter or constrictor of the pupil and the ciliary muscle. And both of those will have clinical significance in a patient that has an oculomotor nerve lesion. The facial nerve parasympathetics exclusively innervate glands, lots of glands. And because it innervates lots of glands, facial nerve parasympathetics need two ganglia to synapse in because of the number of glands that they innervate. Facial nerve parasympathetics innervate two salivary glands, the submandibular and the sublingual, through a synapse in the submandibular ganglion. And through a synapse in the pterygopalatine ganglion, facial nerve parasympathetics innervate all of the oral and nasal mucous glands. The most clinically significant, diagnostically significant gland innervated by facial nerve parasympathetics is the lacrimal gland, because patients with a complete lesion of a facial nerve will tend to present with an eye that's dry and irritated because of loss of lacrimation. Ninth nerve parasympathetics, through a synapse in the otic ganglion, innervate the other salivary gland, the parotid, which in and of itself is not diagnostically significant to a ninth nerve lesion. And then finally, as we've indicated, vagus nerve parasympathetics synapse in a wide array of terminal ganglia. Terminal ganglia are parasympathetic ganglia that are largely found in or near an organ being innervated by vagal nerve fibers in either the thorax or the abdomen. Then the sacral outflow parasympathetics arise from preganglionic parasympathetic neurons at sacral spinal cord levels, specifically S2, S3, and S4. They, like the vagus nerve, synapse in terminal ganglia. And their axons, when they leave sacral spinal cord, run in pelvic splanchnic nerves. And these terminal ganglia, through their pelvic splanchnic nerve innervation, provide a parasympathetic innervation to hindgut viscera and pelvic viscera, including the detrusor muscle of the bladder, erectile tissue, as well as we indicated hindgut structures in the descending colon, rectum, and so on. And then finally, classic congenital malformation known as Hirschsprung's disease results when there are missing terminal ganglia, most commonly in part of the rectum. And this newborn infant then will develop what is known as aganglionic megacolon, where they will not be able to pass meconium because the missing terminal ganglia will not be able to generate peristalsis in the area of the rectum where they are absent. And then in summary, chart illustrates basically the points that we made earlier, and that is we know that there are four cranial nerves that carry preganglionic parasympathetics out of the brainstem. Three of them are linked to four of the cranial parasympathetic ganglia. The facial nerve is serving two of them, and their job, as we've indicated more specifically earlier, is to innervate various glands, salivary glands and others, and smooth muscles, particularly two smooth muscles in the orbit that have a cranial location. Vagus nerve parasympathetics are wandering throughout mainly the thorax and the upper part of the abdomen, synapse and terminal ganglia that we said are in or near the walls of the viscera being innervated, and serve glands and smooth muscle and or cardiac muscle in the heart, the lungs, and the two-thirds of the GI tract. And finally, the pelvic splanchnic nerves arising from S2, 3, 4, also interacting with terminal ganglia, but in the walls of hindgut structures and pelvic structures, including the bladder and the erectile tissue. Now let's contrast the parasympathetic nervous system with the sympathetic nervous system. The first point we want to make in the similar sort of diagram is that the location of preganglionic sympathetic neurons have a thoracolumbar organization, meaning that there's no overlap between where we find preganglionic sympathetic neurons versus preganglionic parasympathetic neurons. So the sympathetic neurons, preganglionic ones, are found continuously, beginning at the T1 spinal cord segment all the way down, and you can see on this diagram, including the L2 spinal cord segment. So there are 14 spinal cord segments, T1 to L2, that contain preganglionic sympathetic neurons. Now, their goal in life is to extend their distribution throughout the body, unlike parasympathetics that have a much more restricted distribution, whereas where we don't find parasympathetics being distributed anywhere to the body wall. Sympathetics have to go everywhere in the body because their major job is to innervate vascular smooth muscle and sweat glands everywhere, vascular smooth muscle including every blood vessel in the body. So there are a wide variety of and more numerous postganglionic neurons that are related to the sympathetic nervous system. And there are two classes of them. One is we find postganglionic sympathetic neurons in the sympathetic chain or trunk of ganglia that are situated in a paravertebral position. Hence the name, these are called paravertebral ganglia. If a preganglionic sympathetic axon does not synapse in a paravertebral ganglion, the axon will pass through that ganglion and will synapse then in a prevertebral sympathetic ganglia. Prevertebral sympathetic ganglia are only found below the diaphragm and are going to participate in the sympathetic innervation of viscera in the abdomen and pelvis specifically. Whereas a general function, as we will see, of the paravertebral ganglia is to serve the body wall by innervating vascular smooth muscle and or sweat glands in or near the body wall respectively. Let's now look at a higher magnification view of just the upper part of the sympathetic pathway and make a couple of points. One is we're looking at the upper part of the thoracolumbar outflow, and we're seeing again examples of preganglionic sympathetic neurons that are leaving from the thoracic spinal cord. Note that all of those preganglionic axons must enter the paravertebral chain of sympathetic ganglia, where they have a couple of choices. They can either synapse at a ganglion cell at their level of point of entry, or the axons can go up to higher levels or down to lower levels and synapse with ganglia above or below their point of entry. The goal, again, of the paravertebral ganglia is to distribute postganglionic sympathetics everywhere in the body to the body wall, serving mainly sweat glands and vascular smooth muscle. All right. And again, we see an illustration that if a preganglionic sympathetic neuron does not synapse in the paravertebral trunk of ganglia, it must pass through that trunk and form a root of a splanchnic nerve. And as a root of a splanchnic nerve, it then must synapse in one of the prevertebral ganglia and participate in the innervation sympathetically of structures in the abdomen or pelvis below the diaphragm. And because of that point, because prevertebral ganglia are only found below the diaphragm and not in the thorax, what this means is that, you can see this by the dashed lines, the thoracic viscera must receive their sympathetic innervation not from prevertebral ganglia, but from paravertebral ganglia. So the paravertebral ganglia that are linked to the upper thoracic outflow from about T1 to T5 serve a dual role. They innervate body wall dermatomes from T1 to T5, but they also participate in the sympathetic innervation of thoracic viscera, including the heart and lungs. The other point that has tremendous significance is a reminder of how the face, scalp, and orbit, the cranial part of the body wall, receive their sympathetic innervation. This is a very important pathway, so let's document the neurons involved and talk about their significance. Number one, the sympathetic innervation of face, scalp, and orbit is one where the preganglionic neuron is found mainly in the T1 spinal cord segment. The preganglionic axon enters the sympathetic trunk, but ascends up to synapse in the ganglion at the top of the trunk, which is called the superior cervical ganglion. And that superior cervical ganglion gives rise to postganglionic sympathetic axons that will run along with branches of the carotid arteries to reach structures in the face, the scalp, and the orbit. Specifically, they will be vascular smooth muscle and sweat glands in the face scalp, and also this T1 to superior cervical ganglion pathway provides a pathway to innervate two smooth muscles in the orbit that are under sympathetic control. One of them is the dilator of the pupil, and the other is the smooth muscle part of the levator palpebrae superioris, known as the superior tarsal muscle. So the next point is, if there's any lesion in a part of the T1 to superior cervical ganglion pathway at those three black arrows that you see illustrated here, the patient will present with the signs and symptoms of a peripheral Horner's syndrome. And as we note on the side of this picture, that will result in an ipsilateral ptosis, a drooping of the eyelid caused by weakness of the superior tarsal muscle, miosis caused by a weakness in the dilator of the pupil, and anhydrosis, a loss of sweating, disrupting the innervation of sweat glands on the corresponding side. But we need now to make an additional point, and that is we've talked up till this moment about how autonomic pathways are two-neuron pathways in the peripheral nervous system. But what we need to point out is also illustrated on this picture, and that is there is a part of the CNS, the hypothalamus, that controls autonomic, particularly sympathetic function. And in order to do that, a neuron cell body in the hypothalamus has to give rise to a bundle of axons that run through the entire length of the brainstem and the cervical spinal cord on their way down to synapse, as we show in this diagram, with preganglionic neurons that are in a thoracolumbar spinal cord location. So the name of this pathway, axons that are running from hypothalamic neurons down to thoracic spinal cord, these axons course in what are known as the hypothalamospinal or the descending hypothalamic fiber pathway. And their job is to allow the hypothalamus to control sympathetic tone. But more to the point, if we have a lesion, and we'll see this in cervical spinal cord lesions and in brainstem lesions, if there is a lesion of this descending hypothalamic fiber pathway, the patient will present with a central Horner's syndrome. So the point is, we can have lesions that are entirely contained within the parts of the brainstem or parts of the spinal cord that will also result in the signs and symptoms of Horner's syndrome, even though the lesion does not affect either the preganglionic or postganglionic sympathetic neuron. Now, picture at the bottom of this chart is enlarged on the next figure. And again, it makes 2 points. One is that certainly one of the jobs of the preganglionic sympathetic neuron, as we've indicated, is to synapse in the paravertebral ganglia and to provide the sympathetic innervation to the body wall. So what you're seeing here indicated by the asterisks are the postganglionic sympathetic axons that will participate in body wall innervation. As we'll see, those dashed lines are termed gray rami when they participate in body wall innervation. The other point that's also made on here, and we're going to expand it briefly now, is that we've said that if a preganglionic sympathetic neuron does not synapse in the sympathetic trunk, it must pass through that trunk and form a root of a thoracic or a lumbar splanchnic nerve. Splanchnic means visceral, meaning that their goal is to synapse in one of the prevertebral ganglia below the diaphragm and participate in the visceral innervation of smooth muscle or glands that are serving gut structures and abdominal pelvic viscera as well. So note that the other important feature about these splanchnic nerves, one is thoracic splanchnic nerves begin at the level of the T5 spinal cord segment. So that thoracic and lumbar splanchnic nerves basically consist of the preganglionic sympathetic axons that arise continuously from the T5 to the L2 spinal cord segment. Whereas from T1 to T5, there's a little bit of an overlap. They're all synapsing in paravertebral ganglia, regardless of whether they are participating in the sympathetic innervation of the body wall, including the face and the scalp, or thoracic viscera, because there are no prevertebral ganglia in the thorax. The other point that's made at the top of the slide is a very important one. Note that Splanchnic nerves, sympathetic splanchnic nerves, carry visceral pain fibers back from thoracic, abdominal, and pelvic viscera. And these visceral pain fibers participate in a phenomenon known as referred pain, where if there's a pathology associated with a thoracic, abdominal, or pelvic visceral structure, because the visceral pain fibers course back with the sympathetic innervation, the visceral pain will be referred to a body wall dermatome that matches the sympathetic innervation of the affected structure. So being general about it, when patients have referred, poorly localized, dull, aching pain over the T1 to T5 dermatomes, that is a classic indication of a thoracic problem, commonly angina pectoris or coronary artery disease, but it could be other thoracic issues as well. If a patient presents with referred pain over the T5 to L1 or L2 dermatomes, that's a classic indication of some sort of pathology associated with an abdominal or a pelvic visceral structure, respectively. Finally, let's use this chart to summarize the details of the sympathetic or thoracolumbar outflow. As we indicated in the previous diagrams, that all of the preganglionic sympathetic neurons are found continuously from the T1 to L2 spinal cord segments, and that they have a choice. They can either synapse on paravertebral ganglia and participate in body wall innervation, or synapse in prevertebral ganglia and participate in the sympathetic innervation of viscera below the diaphragm. So we know, generally speaking, that the paravertebral ganglia are serving glands and smooth muscle in the body wall, including the clinically significant T1 to superior cervical ganglion face scalp orbit pathway. And we know that prevertebral ganglia are only found below the diaphragm. Therefore, the paravertebral ganglia, in addition to serving the body wall, must also, as we say in the chart, provide the sympathetic innervation to thoracic viscera. And then, as we indicated, the thoracic splanchnic and lumbar splanchnic nerves are preganglionic sympathetic axons that pass through the paravertebral ganglia and therefore synapse in the prevertebral ganglia below the diaphragm and participate in the innervation of those visceral structures. And lastly, we said that it is those splanchnic nerves that carry visceral pain fibers back from not only abdominal pelvic viscera, but from thoracic viscera, and play a significant role in the phenomenon of referred pain, where the dermatomal localization of the referred pain matches the sympathetic innervation of the visceral structure in which the pathology is generated. Final diagram regarding sympathetic outflow is just some details about nomenclature. This is a cross-section of the spinal cord somewhere between T1 and L2 and indicates the specific location in the gray matter of the spinal cord where preganglionic sympathetic neurons are found. They're found specifically in the lateral horn from T1 to L2, and since they are motor axons, all of the preganglionic sympathetic axons must leave in the ventral root of a corresponding spinal nerve from those segments. And as we know, all of the preganglionic axons will join briefly with the spinal nerve, course briefly with the ventral ramus, and then all of the preganglionic axons, as we've alluded to earlier, must leave the spinal nerve, ventral ramus, and enter the paravertebral trunk of sympathetic ganglia. The term that describes the course taken by a preganglionic sympathetic axon from the ventral ramus to the paravertebral ganglion is known as a white ramus. So a white ramus is a myelinated preganglionic sympathetic axon arising anywhere between T1 and L2. So there are 14 of those. Then, one of the options that white ramus has, which is illustrated on this slide, is to synapse in that paravertebral ganglion. Then as such, the postganglionic neuron in that ganglia may participate in body wall innervation. If it does, then the postganglionic axon that leaves a paravertebral ganglion cell is known as a gray ramus. It is an unmyelinated postganglionic sympathetic axon that will then rejoin the corresponding spinal nerve and course with all of the branches of the dorsal and ventral rami to reach glands and smooth muscle in, obviously, the body wall and the limbs. So in terms of terminology, there are actually 31 pairs of gray rami, because the gray rami must rejoin all of the 31 pairs of spinal nerves and be distributed in a dermatomal fashion that corresponds to the area of the body wall innervation.